This is Mr. Maxwell for Pingry AP Biology. Today I want to talk about some aspects of metabolism. First, I want to talk about, as an overview, if we take a plant cell, and this is kind of a representation of a plant cell. It's missing a cell wall, but it'll do. There are basically three big compartments that we want to talk about at the AP Biology level. As far as what happens in metabolism. Metabolism is exceedingly complex, and realize that we're only picking a couple of pathways to look at. So first, this video we're going to talk about glycolysis and fermentation. In another one I'll talk about respiration and then photosynthesis, but I just include this as an overview slide. So glycolysis and fermentation happen in the cytoplasm, and obviously respiration happens in the mitochondria, photosynthesis in the chloroplast. And so we're going to focus on glycolysis first. And so if we look at the process itself, you should be able to tell from metabolic pathways, you should be able to look at these and figure out what's going on. That's the most important thing to get out of any of this, is to not be frightened when you look at metabolic pathways. So first, you should notice each one of these arrows is is a reaction that's catalyzed by an enzyme. So anytime you see an arrow, you know there's an enzyme. You should also notice that here we have ATP being hydrolyzed to ADP, and you should recognize that reaction as a downhill reaction. And typically, if we're using ATP, we're driving an uphill reaction. So this reaction adds a phosphate onto glucose, and that's the first step in glycolysis. And so we're actually putting energy into the system. We're expending some ATP to make this product. And then we expend another ATP. So overall, for every glucose that we enter into this, we're gonna invest 2 ATPs, and the books will call this the investment phase of glycolysis. Another point to realize is that while we're showing one particular pathway, you know that fructose is also an energy source and can be fed in here, and glucose can be produced from starch, and so there's complex things that get fed into this. But we're just showing one set of reactions. So once we get this compound here, fructose 1,6-bisphosphate, ready to go, we've essentially pushed the molecule up an energy hill so that it's ready to fall off the precipice. And in this reaction, this compound gets chopped into 2 3-carbon compounds. So it's a 6-carbon compound chopped into 2 3-carbon compounds. And then this glyceraldehyde 3-phosphate is the compound that gets drawn off and used in the rest of the pathway. This dihydroxyacetone phosphate is actually just an isomer of glyceraldehyde 3-phosphate, and you can see that they're very similar compounds. The only difference really is where this double-bonded oxygen is, and there's an enzyme that catalyzes this exchange. And if you remember Le Chatelier's principle from chemistry, you'll know that if you draw off a particular reactant, it will pull a reaction in the direction of the thing that you're pulling off. And so even though dihydroxyacetone phosphate isn't directly used downstream, it gets pulled into this form and then used downstream. So the upshot of this, though, is that you start off with glucose, a 6-carbon compound, and then you make 2 3-carbon compounds, and you use some ATP. And then in the next step, you're going to take these 2 G3Ps that you have from that previous process, and you're going to harvest some electrons. Notice NAD plus is an electron carrier, and NADH is, NAD plus gets converted to NADH, gets reduced to NADH, and in the process picks up some electrons. And then if you have an electron transport chain in the mitochondria, you can regenerate NAD plus by harvesting those electrons and using them um, to do energy processes. But that's a later step. Then you see that in these reactions, we harvest an ATP, and because this happens twice, because you have 2 3-carbon compounds here, you get 2 ATPs back. So you're even. You invested 2, you got 2 back, and then down in this step here, to make pyruvate, you wind up getting 2 more ATPs back, 1 per reaction. So that's basically the steps of glycolysis. There's no need to memorize any of this stuff, but you should be able to analyze any pathway you get and be able to tell the important things what's going on. So once you have pyruvate, there are 3 fates of pyruvate, one of which is to send the pyruvate to the mitochondria and do aerobic oxidation of it. The other 2 steps are fermentation steps, or fermentation pathways, one of which happens in humans where we make lactate, and you've probably heard it called lactic acid, or in yeast, for example, they produce ethanol. And in the process, give off a CO2. The whole point, though, of fermentation is that we have this NADH, and if we don't produce NAD+, we can't continue to do glycolysis. So those electrons that have been placed on NADH need someplace to go. If you don't have a mitochondria, you can put them on pyruvate and convert it to lactate, which you can later turn back into pyruvate if you have the ability to do respiration. But this NAD plus is vital because you can't continue to do glycolysis unless you have that. And likewise, alcoholic fermentation also regenerates NAD plus. It's the whole point of it. Ethanolic fermentation really isn't reversible because you've lost this CO2, whereas lactic acid fermentation, at least in our bodies, can be reversed. So the next step would be respiration, and I'm going to break this here and do another screencast about respiration. But that's the important stuff that you need to know about glycolysis and fermentation. So this is the step here that I was referring to that you have to have fermentation for so that you can get this NAD plus back.